What makes antibiotics only target bacteria, not human cells in sickle cell? Imagine trying to fix a broken car engine without damaging the rest of the vehicle. That's similar to how antibiotics work in our bodies. They are designed to attack bacteria specifically, leaving our human cells unharmed. This selectivity is crucial, especially for people with sickle cell disease who are more prone to infections. The main reason antibiotics target bacteria and not human cells lies in the fundamental differences between the two. Bacteria have unique cell structures that human cells do not. For example, bacteria have a cell wall made of peptidoglycan, a substance human cells lack entirely. Antibiotics like penicillin and other beta-lactams interfere with the production of this cell wall, causing the bacteria to weaken and eventually die. Since human cells do not have cell walls, these antibiotics do not affect them. Bacteria also have different membranes and envelope structures. Some antibiotics, such as polymyxins, work by disrupting bacterial membranes specifically because their composition is different from human cell membranes. This difference allows these drugs to target bacteria without harming our own cells. Another key difference is in how bacteria make proteins. Bacteria use 70S ribosomes for protein synthesis, while human cells use 80S ribosomes. Antibiotics like tetracyclines, macrolides, and aminoglycosides bind to bacterial ribosomal subunits, blocking bacterial protein production. Since human ribosomes are different, these antibiotics do not interfere with our cells' protein-making process. Bacteria also have unique enzymes involved in DNA replication and other vital processes. For example, fluoroquinolones target bacterial enzymes called DNA gyrase and topoisomerase Roman 4, which are essential for bacterial DNA copying. These enzymes are either absent or structurally different in human cells, so the antibiotics do not affect our DNA processes. Additionally, some antibiotics block bacterial pathways for making folic acid, a vitamin humans get from food. Since humans do not produce folic acid themselves, these drugs specifically inhibit bacterial metabolism. In the context of sickle cell disease, this selectivity is especially important. Patients with sickle cell often have weakened immune systems and impaired spleen function, making them more vulnerable to infections. Antibiotics can help prevent or treat these infections effectively without damaging their own cells. This targeted action supports safe and effective infection control, which is vital for managing health in sickle cell disease. In summary, antibiotics are able to selectively target bacteria because they exploit differences in cell wall composition, ribosomal structure, and specific enzymes involved in DNA replication and metabolism. These differences allow antibiotics to kill or inhibit bacteria without harming human cells, making them a powerful tool for infection prevention and treatment, especially in vulnerable populations like those with sickle cell disease. Always remember to follow medical advice and consult healthcare professionals for personalized care.